Hello, check. Hello, namaskar, and a very good morning, ladies and gentlemen. I'm your Sutrada Ridhima Majumdar, welcoming a very warm welcome to all of you. So this day is really very special for us as we have the inaugural day, as well as we also have the technical session for all of us. So at this point of time, we all gonna keep our eyes sharp on the screen as well as ears keen with the speakers and with the dignitaries sitting over here with their thoughts and wisdom and the words of wisdom. So yes, let's begin this wonderful morning with all of us. So following the success of Skilling Biotalent, SBT-2025, dated 7th March 2025, sponsored by Gujarat State Biotechnology Mission, GSBTM, which highlighted opportunities, employability skill, internship pathway, and entrepreneurial possibilities for life science students under the vision of NEP 2020. The proposed SBT 2.0 2026, dated March 16, 2026, aims to advance this journey from awareness to actionable capacity building in research and innovation-driven careers. While the previous edition focused on job compliances in pharmaceutical, agriculture, microbiology, and molecular biology sectors and exposed participants to start-up and entrepreneurship possibilities, the upcoming symposium is designed to deepen developmental skill essential for research-oriented, R&D, and industry-aligned career trajectories. Despite expanding opportunities in India's biotechnology sector, a persistent mismatch between the academic training and industrial expectations continues to limit the employability of life science graduates and leaves many industry positions inadequately filled. Academic research frequently remains focused on publications with limited translation or market-oriented outcomes, while industry struggles to identify candidates with hands-on problem-solving abilities, regulatory and quality compliance awareness, and the capacity to work in interdisciplinary collaborative R&D environments. In this context, the one-day National Symposia will deliberately address these gaps by emphasizing competences in research proposal writing, understanding the funding and the innovation ecosystem, identifying industrial relevant research questions, and aligning academic projects with real-world and R&D needs. The program will feature two focused keynote talks followed by a panel discussion involving experts from academia and industries, enabling an open dialogue on recruitment challenges, skill gaps, and practical strategies to enhance students' readiness for industrial roles and research careers. This initiative is sponsored by GSBTM Gandhinagar. Heading up with a small little profound introduction of GSBTM, the biotechnology is a multidisciplinary field that utilizes living organisms, cells, or their components to develop products and processes that improves human life and the environment. It combines biology science with engineering to solve problems in healthcare, agriculture, and industry. It used to produce pharmaceutical like insulin, develop disease-resistant crops, create biofuels, and for environmental waste treatments. Core methods include genetic engineering, molecular diagnostics, and enzyme technology. Hence, let's hear a huge round of applause for this day and for ourselves, wherein we are beginning this wonderful day, not just with the knowledge, but also with the gaining appreciation, where we are sitting over here and we are here to gain the acknowledgement by our speakers and dignitaries. So let's hear a huge round of applause. Now, moving ahead, before I start this day, I would like to invite our dignitaries to the lamp lighting ceremony, a timeless tradition that symbolizes wisdom, positivity, and the trump of light. The lighting of the lamp signifies the awakening of knowledge, the removal of ignorance, and the beginning of the journey filled with hope and success. Now I request everybody to please come forward.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपो हरतु मे पापम् सन्ध्यादीपनमोऽस्तुते ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः
Let us put our hands together, please, initiating this wonderful day, seeking the blessings.
रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् यस्य दृष्टे गृहे दीपः तस्य नास्ति दरिद्रता शुभं कुरुत्.
Hereby, I would also like to play the university song, and I request each one of you to please mark up their presence over here by raising a bow for the university song.
धन्य धन्य धरती मंगल दक्षिण गुजरात स्थापित विद्यापीठ हमारी रम्य अनेरड़िया तापी पूर्णा सरित नर्मदा अंबिका कावेरी लाल सदाए लीलिछम नदियों न पटकुल पहरी धन्य धन्य धरती लीली मंगल दक्षिण गुजरात स्थापित विद्यापीठ हमारी रम्य अनेरड़िया पश्चिमी रत्नाकर तेरी पूर्वे तरंगमान कुदरत नीचे महर के भूमि बारे मास रसार सरल प्रकृति ना सौलो को आनंदी अभिजार धन्य धन्य धरती मंगल दक्षिण गुजरात स्थापित विद्यापीठ हमारी रम्य अनेरड़िया
दक्षिण गुर्जर क्षेत्रनी विद्यापीठ अमारी माता
दक्षिण गुर्जर क्षेत्रनी विद्यापीठ हमारी माता सत्य ज्ञान ने अनंतनी एच उज्ज्वल उदिगाता तेना गौरव प्रगति विकास हमने प्राणती वाला एने कंठे पहरा विशु सिद्धिनी जयमाला 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 विद्याने चरण धरिए निष्ठाना परिजा धन्य धन्य धरती लीली
Kindly take down your seats, please. Now moving ahead, we would like to felicitate our dignitaries present over here. So I request Dr. Gaurav Shah, Symposium Coordinator, Professor and Head of Department of Biotechnology, to please come forward to do the honors. So we would like to felicitate Dr. Apurva Desai, Director, IQAC, Surat, with a flower bouquet, memento, and a calendar from MBSI, Microbiology Society India, scientific organization dedicated to advancing microbiology through education, research, and collaboration. Next, we would like to felicitate Dr. Narendra Bhai Patel, in charge Registrar VNSGU Surat, and I request Dr. Gaurav Shah to do the honors. And once again, I would request Dr. Gaurav Shah to come forward and do the honors of Shri Rahul Kharadi, Deputy Manager, GSBTM. And now I request Dr. Nikunj Tandel to come forward, the Organizing Secretary of SBT 2.0, and do the honors of Dr. Gaurav Shah, Symposium Coordinator, Professor and Head of the Department of Biotechnology. Now, moving ahead, I would like to call upon the podium Dr. Gaurav Shah, Symposium Coordinator 2.0, Professor and Head of Department of Biotechnology, for his welcome address. Good morning, sir.
Thank you. Namaste. Good morning, all of you. Honorable Dr. Apurva Desai, sir, IQAC Director of Veer Narmad South Gujarat University. Dr. Narendra Patel, sir, in charge Registrar of Veer Narmad South Gujarat University. My friend from GSBTM, Deputy Manager Shri Rahul Kharadi. Dignitaries of the dais, teachers, students, and my dear friends. Skilling biotech talent, development of skill, considering National Education Policy 2020, was the idea which we introduced last year and was acknowledged by Gujarat State Biotechnology Mission. Considering that what are the internship opportunities and job prospects for the students. After that remarkable success of that event, GSBTM again sponsored with association with Skill Biotech 2.0. 2.0 ને અત્યારે પેલું ચાલે છે ટ્રેન્ડ, કોઈ પણ એક હોય પછી 2, પછી 3, something like that. So નામ what we thought we kept same name, Skill Biotech Talent 2.0. What we are going to discuss today, we have on this platform, I am very much fortunate enough that representations from 10 universities and 15 industries have agreed to be a part of this event. And that is all for you, students, the future of our nation. The idea is that how to fill that gap. Industry has always a complaint that whatever the university produces are not industry ready. And academics and universities have their own limitations also. So, Government of Gujarat, Gujarat State Biotechnology Mission took these initiatives and given this responsibility to us that why not to discuss this, the limitations of academics, limitations of the industries, and how we can bridge this particular gap. Skill Biotech Talent 2.02 is all about research and how that research not restricted to the laboratory, but how it can be commercialized. Development of skill, development of entrepreneurship, generating job, being independent, self-reliant, Make in India is the only way out for all of us. And biological scientists, those who are associated with biology, and obviously integration with artificial intelligence, computer science, and information technology will be the next leap in case of human evolution. So I request all the students to actively participate. The first part of this symposia is regarding research opportunities and translation of research, because whatever the hard work you are doing in your laboratory, if it is not beneficial for the society, lot of resources are wasted. And Government of India, Gujarat government wants that you should come up, and industry should be the hand holding. Whatever the limitations at your end, that also you can just put forward today's in second part, that is panel discussion, where all the resource persons from industry, from academy, I call it as galaxy of intellectuals, will try to answer your questions. So your active participation is very much essential. Apurva sir, we have participants from UG NEP. Most important is research scholars and PG who are doing research at the dissertation or at the end of the level, and we have teachers also as a participant, plus even citizens of the Surat city are invited to be a part of it, that what this particular university do, how we channelize things. But finally, it's all on you all, that how you take it and how you take maximum advantage of what Government of Gujarat, Gujarat State Biotechnology Mission is doing, and what other universities, all teachers are doing hard to build our nation in a very effective way. I once again thank on behalf of Veer Narmad South Gujarat University to Gujarat State Biotechnology Mission, Jadeja sir, Snehal Bakhtaria sir, Mitali madam, and Rahul, who had took so much pain so that we intellectual and students can discuss and try to find out some way out, and government is determined to help us. So with that note, thank you so much. Once again, welcome to this campus. I hope this symposia, a day-long investment of your quality time, will lead to some fruitful results, and we will make it sure that we will show that to the state as well as nation. Thank you.
Thank you so much, sir. And now it's time for opening remarks on need of bridging industry academia gap by Shri Rahul Kharadi, Deputy Manager, GSBTM. So let us all put our hands together for him, please. Welcome, sir.
Dignitaries on the dais, dignitary of the dais, faculty members, representative for industries અને વિદ્યાર્થી મિત્રો, GSBTM દ્વારા આયોજિત આ એક દિવસીય સિમ્પોઝિયા જે આપણે VNHUના ડિપાર્ટમેન્ટ ઓફ બાયોટેકનોલોજીના સહયોગથી કરી રહ્યા છે, તેમાં આપ સૌનું હું હાર્દિક સ્વાગત કરું છું. આજના સમયમાં બાયોટેકનોલોજી એ વન ઓફ ધ ફાસ્ટેસ્ટ ગ્રોઈંગ ફિલ્ડ છે, જે એગ્રીકલ્ચર, હેલ્થકેર, ફાર્મા અને એન્વાયરમેન્ટ જેવા સેક્ટરમાં રિવોલ્યુશનરી ચેન્જીસ લાવી રહ્યું છે. GSBTM રાજ્યમાં રિસર્ચ ફંડિંગ, ઇન્ડસ્ટ્રી પોલિસી સપોર્ટ ઉપરાંત વિવિધ ટ્રેનિંગ પ્રોગ્રામ, FDP સ્ટુડન્ટ કેપેસિટી બિલ્ડિંગ પ્રોગ્રામ જેવા મહત્ત્વના સ્કીમો રન કરી રહી છે. આજના કાર્યક્રમનો ઉદ્દેશ ઇન્ડસ્ટ્રી અને એકેડેમિયા વચ્ચેનો જે ગેપ છે એ ફિલ કરવાનો છે. અંતમાં હું ફરી એકવાર તમામ ડિગ્નેટરીઝ, સ્પીકર્સ અને ઉપસ્થિત આપ સૌનું હાર્દિક સ્વાગત કરું છું. થેન્ક યુ.
And now I would like to invite on podium Dr. Narendra Bhai Patel, in charge registrar, VNSGU Surat, to mark his address.
नमस्कार एंड वेरी गुड मॉर्निंग. मंच पर उपस्थित IQAC डायरेक्टर डॉक्टर अपूर्व देसाई साहेब, बायोटेक कोऑर्डिनेटर डॉक्टर गौरव शाह सर અને GSBTMના ડેપ્યુટી મેનેજર ડૉક્ટર શ્રી રાહુલભાઈ. આજના જે નેશનલ સિમ્પોઝિયા છે, સૌપ્રથમ તો હું ગૌરવ શાહને ખૂબ ખૂબ અભિનંદન પાઠવું છું કે એમણે કીધું છે કે 2 નંબર આવી ગયો છે. હું એમને એવા અભિનંદન આપું છું કે એમની રિટાયરમેન્ટ એજ થાય એટલા એ લોકો રિટાયરમેન્ટ એજ સમયે પણ એ નંબર આવી જાય. એટલે આવા સિમ્પોઝિયા તમે કન્ટિન્યુઅસ અથવા બીજા કોઈ પણ પ્રોગ્રામ કરતા રહો. આવી અમારા વીર નર્મદ સાઉથ ગુજરાત તરફથી ખૂબ ખૂબ અભિનંદન પાઠવું છું અને બેસ્ટ ઓફ લક કહું છું. ઓફ ધ ડાયસ વિવિધ કોલેજોના સ્ટાફ મિત્રો, એટલે કે ટીચિંગ સ્ટાફ અને વિદ્યાર્થી મિત્રો, ટૉપિક ઘણો સરસ છે અને ગૌરવ સરે ઓલરેડી કીધું છે કે રિસર્ચ બેઝ પર આ ટૉપિક સિલેક્ટ કર્યો છે અને GSBTMએ પણ આ બાબતને મંજૂરી આપી છે. તો વિદ્યાર્થી મિત્રોને એટલી જ સલાહ પૂછો કે તમે આજે જે પણ કંઈક થોડું તમારા સબ્જેક્ટ રિલેટેડ કંઈક ભાથું લઈ જજો અને ભવિષ્યમાં આ જ વિષય ઉપર, એટલે કે રિસર્ચમાં તમારે જે મનગમતો વિષય છે એમાં કન્ટિન્યુઅસ રિસર્ચ કરજો. બસ એવી શુભેચ્છા. થેન્ક યુ વેરી મચ.
Now, moving ahead, I would like to call upon the podium Dr. Nikunj Tandale, Organizing Secretary for SBT 2.0, as we are here right now at the point where we'll be having unveiling of information booklet by our dignitaries. But before that, I request him to please come forward on the podium.
ચાલુ છે ફૂલ સ્ક્રીન હું કરી દઉં છું, પછી હું ખાલી કહેજો એનું વ્રત હું કરાવી દઉં. ઓકે.
આતિથ્ય દેવો ભવ આપણી ભારતીય સંસ્કૃતિને માનમાં સૌથી ઉચ્ચતમ ગણવામાં આવે છે અને અમારા ભાવભર્યા આમંત્રણ આપીને અત્રે ઉપસ્થિત અમારા સૌ કોઈ, મારા માટે તો સૌ આદરણીય જ છે કારણ કે હું બહુ યંગ છું, તો અમારા આમંત્રણને માન આપીને અત્રે ઉપસ્થિત સર્વ ગુરુજનોને મારા આદરસર પ્રણામ. સૌપ્રથમ મારા વિદ્યાર્થીગણોને પણ મારા ખૂબ ખૂબ અભિનંદન કે તે લોકો આવ્યા છે. ઓનરેબલ વાઇસ ચાન્સેલર સર, respected registrar sir, deputy registrar sir, IQSC director sir, as well as from GSBTM sir, all the representative who all present over here, I welcome you all. મને હજુ પણ યાદ છે જ્યારે ફર્સ્ટ મિટિંગ થઈ હતી સર જોડે, તો સરનું એક વાક્ય હતું કે નિકુંજ મારે કંઈક આપવું છે કે ઇટ શુડ બી ઇન એ સ્મોલ પોકેટ લાઇક બુક કે જે લોકો કેરી કરીને લઈ જઈ શકે એમ કરીને. તો એટ દેટ મોમેન્ટ ઓનલી આઈ હેવ અ થોટ કે વોટ શુડ આઈ ગીવ, because I had experience of entire India, most probably doing my PhD and postdoc. That specifically, of course, right now our Gujarat is leading. But when we need to talk about that, how much students are aware about the different examinations as well as fellowship. So I told immediately, sir, that I have an idea in my mind. So that idea, along with my team who are standing over there, so Mr. Jainil from MSc Biotechnology and all the young girls from semester 4 microbiology, so huge round of applause for them. They have helped me in finalizing this book actually. So we are inaugurating one book which will be given to all of you as a soft copy. So I'll just inaugurate with the permissions of Apurva sir as well as all the dignities. Sir, Apurva sir, please come sir. हाँ, पेला में देखा. So this is the book what we are going to give you in a soft copy. I'll just give you the glimpse of it. So of course the message is from our Vice Chancellor sir. Then what we are trying to cover for all of you is, of course, first of all we'll be discussing about GSBTM policy. It's a huge policy, so of course with the help of my students I've summarized in 10 pages, and with each content there is an infograph of it. So students, if you do not want to read all those pages, they can directly go to the infograph and they can get the glimpse of it. Second point is, after BSc, what all exams are there at the master's levels throughout the India, when they are conducting the exams, what mode of examination is there, every information is available over here. Followed by, after masters, different options such as JRF, SRF, PhD, postdoc, all the fellowships in India, when it open, when the calls comes, what all the basic requirements are there, along with how much fellowship amount you can get it or you can claim it. Every informations are there. And we are sitting in a Gujarat state who's always lead in the form of startup. So at the end, what we are giving is that how many startups ideas you can go through it, which you can deliver to the government platforms. So across the India, how many bio startups institutions are there of governments, and when the open calls has been come, that has been given in that book. I'm just giving the glimpse that what all the things are that. This is the GSBTM policy about what GSBTM is actually doing for this state specifically. This slide is specifically for the students who are doing internships over there, as well as for my young faculties. Many of you know that research scheme is going on RSS, and every alternate month the schemes has come. So through this you can get to know about when the calls is open and all. Then this is the summary of all JRF, SRF, PhD and postdoctoral fellowships. Of course details are there on each and every page, you can get it. And at the last is different startups policies in India. So I hope that what I didn't understand at my young age, definitely the young minds who are sitting back, they will understand. And at the end, definitely I would like to thank my another organizing committee member, Dr. Ambika ma'am, my fellow colleagues, Dr. Chirag sir, Milan sir, Saddam sir, and Mihir sir, who at the end done the proofreading of this. And of course, Dr. Gaurav Shah sir, who has given the approval that yes, we can go ahead with this. Once again with this, thank you all of you.
And after this wonderful unveiling of the information booklet by our dignitaries, it's time for our inaugural address by Dr. Apurva Desai, Director IQAC, Surat. Good morning, sir. Thank you, sir.
Good morning, everyone. Good morning. Special on the back. Good morning. So, very good morning to all of you. I'll start with, addressing the students first. So, my dear students, then my colleagues who have come from various universities, various colleges of our state, colleagues from other departments, colleagues who is there on the dais, ladies and gentlemen. I'm not the right person to speak to you because I'm not having, any knowledge about biotechnology. I do not know what is the correct spelling of biotechnology, whether it starts from B or not, that I don't know. So I'm probably not a correct person or right person to speak to you. But as I am, carrying responsibilities of IQSU and I am invited here, it is my moral responsibility to talk to you regarding something, So first of all, I am very much delighted and I congratulate Dr. Gaurav Shah, the professor and head of Biotechnology department, for inviting me and giving me this honor. And I congratulate him for organizing such a beautiful gathering event. And in fact, Biotechnology department is one of our flagship department. It is very vibrant department. Every now and then they are busy with some events. And sometimes I get upset with Gaurav also. Why do you, organize so many events and not giving time to us? And I hope he knows what I'm referring to. But this is our, first and very important responsibility to do events for our students. I also congratulate the entire team of the students especially for, developing such a beautiful booklet, very informative one. And it is very much useful for our students. So I congratulate the department for that too. Now, this is a time where basic sciences is, struggling. And young students are finding some other opportunities and possibilities. And one of the possibilities was, intentionally I am using was computer science. I am a person of computer science, And most of the people were going in the field of computer science for last 2 and 2.5 decades. But we are witnessing a transition these days. The core computer science is, struggling a bit nowadays. And this is a time, where some other options are required for the students and for the young brain. In fact, a computer science department in our university, computer science department took an initiative to start a bioinformatics program 15 years back, 12, 15 years back. And Chirag is a student of that batch, first batch of bioinformatics student. We took initiative. And now this responsibility lies with the biotechnology department, bioscience department to carry it forward. This is a time where you can take initiative in bioinformatics and biotechnology. I am sure that the Department of Biotechnology will take up this initiative. Biotechnology, in between, Gaurav, referred to something. Biotechnology, skilling biotech talent 2.0. Where this 2.0 is come from, 0.0 or 2.0 come from, It is, contribution of computer science. In fact, we manage versions, version 1, version 2, version 3, and that 2.0 is version 2.0. So in our day-to-day life, we have adopted different fields in our communication, in our, so many things. Here it is an example where you have adopted something from computer science. In fact, we are one computer scientist or computer science people, they are one who have borrowed a lot many things from biosciences. For example, genetic algorithms. We have adopted genetic engineering from biotechnology. The male and female mating, and it generates the third child, and that concept we have adopted, and we have evolved some new theory and algorithms. Neural networks is another where we have adopted biosciences, and we have grown. My question is, when you are talking about research, my question is, the same thing you cannot do. We all people, generally this is a very bad situation in probably our part of world. We want to hide our work from one another. What I am doing, મારે એમને નહીં કહેવું જોઈએ, otherwise he will steal my idea and she will excel. Now this is not the correct approach. In the era of interdisciplinary and multidisciplinary, we should exchange our ideas. In fact, why, Gaurav, you have invited only students of biotechnology and biosciences? In fact, you could have invited students from other faculties like computer science, where you can give one platform to students to interact each other's ideas. And that is probably the perfect start for collaborative and multidisciplinary research, right? So 3.0. So go for 3.0 and invite all the students for 3.0 in that. For that matter, you should invite all science students. All science students you can invite, and you can give a platform to interact with one another. So this is my small request to you. The second thing is, you are doing so much of research, so much of practical. I know some paper making project you took up last year, probably. Some sand brick or the plastic brick project you are doing this year. As an IQSC director, I should know all the things. I'm not a spy, but I come to know about these things. The question here is, when you are doing so much of research, why you cannot commercialize it? That is the most important thing. Your students are also doing so much of research, so much of projects. So how we can commercialize it? My small request to all the students is, whatever research that you do, the first thing that you should do is try to file for patent. ગૌરવ, બધા જ પ્રોજેક્ટ, જેટલા પ્રોજેક્ટ હોય એ બધા પ્રોજેક્ટને પેટન્ટ બનાવવા માટેની એક પ્રપોઝલ we have to submit. That is what I am doing in my department. All the AIML students, they have to submit patent proposal to the university. Compulsory. Not today, not tomorrow, maybe day after tomorrow, some student will get, patent. So that is where commercialization can come in picture. If not commercialized, if not accepted as a patent registration, you should go for publish a good paper. Now, why this patent writing and paper writing is important for you as a student? Now, if you write a good paper, if you write a good patent, then you can reach to so many people in the world, and that will become, a channel, communication channel between you as a entrepreneur, young researcher, with rest of the world. And that is one channel from where you can get so many, you can unwind or you can uncover so many possibilities. You never know. The world is, in search of so many solutions to so many small problems. I share with you one simple thing, my experience, one of my experiences. One of my student, we PhD student, we published one paper in the international journal. And that paper was on online character recognition. Online character recognition paper was there. To our surprise, after publishing that paper, after 6 or 7 months, somebody from Mumbai approached us. And he was into a business of smart board, where he was looking for Gujarati OCR, online Gujarati OCR, similar to one which we published as a paper. And then we tied up with him, and that project was commercialized. So it is not only, one output, but it is probably one career output or career benefit for you as well. So whatever you do as a research work, you should be, very much you should be into publishing that research work in the journal or patent or something like that. In Gujarat, I always say that the people are in South Gujarat, rather, not Gujarat, South Gujarat. The South Gujarati people, we are very much shy of, glorify ourselves. But the smallest thing that you do in your life, that should be communicated to the world, and let the world know what you are doing. Even the smallest thing, we don't know. Your smallest thing is probably one of the things which solve a big problem for others. So I'm sure that this type of events, and today this event, I am told by Gaurav Shah that in the afternoon, industrialists are coming and they are interacting with you. Take maximum benefit of this type of interaction. They are stalwarts in their own fields. They have got a huge experience. They know how to convert your small ideas into big business. So interact, take maximum benefit of their presence. And I'm sure that this event will be very much fruitful for all of us. And I'm sure that you will take away some good things and good memory with you. Thank you very much.
Thank you so much, sir. And after his words, we also have a beautiful message from Honorable Vice Chancellor Dr. Kishore Singh Chavda. That's going to be a visual message for all of us. And he has sent the best wishes for the symposium for all of us. So let's have his message.
સૌને પ્રણામ. હું ડૉક્ટર કિશોર સિંહ એન ચાવડા, કુલપતિ શ્રી વીરનગર દક્ષિણ ગુજરાત યુનિવર્સિટી, સુરત, ગુજરાત, ભારત. આપણી યુનિવર્સિટી આપણું ગૌરવ. આપણી યુનિવર્સિટી માન્ય પ્રધાનમંત્રી શ્રી નરેન્દ્રભાઈ મોદી સાહેબના નેતૃત્વમાં વિકસિત ભારત 2047 ના સંદર્ભમાં આપણા યુનિવર્સિટીના મોરપીચ સમાન બાયોટેક વિભાગ અને તેના આદરણીય વિભાગીય વડાશ્રી, જેમનું નામ જ ગૌરવભાઈ છે, એવા આદરણીય ડૉક્ટર ગૌરવભાઈ શાહજી, અમારા IQACના વડા અને કમ્પ્યુટર સાયન્સ વિભાગના વડા એવા આદરણીય છે ડૉક્ટર અપૂર્વભાઈ, તમામ એક્ઝિક્યુટિવ કાઉન્સિલ, બોર્ડ ઓફ મેનેજમેન્ટ અને તમામ આદરણીય મહેમાનો અને સર્વ પાર્ટિસિપેટોને વીરનગર દક્ષિણ ગુજરાત યુનિવર્સિટી તરફથી સૌને પ્રણામ. આજે જ્યારે આપણે સ્કિલિંગ બાયોટેક ટેલેન્ટ 2.0 SBT-2026 એ નેશનલ સિમ્પોઝિયમ્સ ઓફ એકેડેમિયા રિસર્ચ એન્ડ ઇન્ડસ્ટ્રી અંડર NAP 2020 જ્યારે યોજાઈ રહ્યું છે, ત્યારે આપણે સૌ જાણીએ છીએ કે વિશ્વમાં પર્યાવરણીય સમસ્યાઓ જે છે, સાથે સાથે આપણે જ્યારે યુવા ભારત તરીકે અને રાષ્ટ્રમાં જ્યારે 50% કરતાં વધારે 35 વર્ષથી ઓછી વયના જે લોકો અહીંયા આપ સૌ બેઠા છે, એઓને અમારી નમ્ર અપીલ છે કે આપણે આવી ટેલેન્ટ જે સ્પર્ધાઓ જે છે એમાં આપણે સૌ ભાગ લઈએ, રાષ્ટ્રના વિકાસમાં વધારે ને વધારે મહત્તમ ફાળો આપીએ અને સાથે સાથે સમગ્ર બાયોટેક વિભાગ અને એના તમામ અમારા શૈક્ષણિક વહીવટી કર્મચારીશ્રીઓ, વ્યક્તિઓ અને તમામ વિદ્યાર્થીઓને આ આયોજન સફળતાપૂર્વક થાય એને માટે હું સૌને શુભેચ્છાઓ પાઠવું છું અને પુનઃ એકવાર રાષ્ટ્રના વિકાસના નિર્માણ માટે જે કંઈ આપણી પર્યાવરણીય વ્યવસ્થાઓ જે છે એમાં વધારે ને વધારે સુધારો થાય એને માટે આપણે સૌ મન, વચન અને કર્મથી કટિબદ્ધ થઈએ અને વસુદેવ કુટુંબકમની વન વર્લ્ડ વન ફેમિલીના સૂત્રને ચારિતાર્થ કરીએ. પુનઃ એકવાર સર્વનો આભાર. વંદે માતરમ. ભારત માતા કી જય. જય જય ગરવી ગુજરાત. આપ સૌને પ્રણામ. હું Dr. Kishore C.
And after his warm wishes,
Gujbati
it's time for vote of thanks by Dr. Ambika Arketkar, Organizing Secretary, SBT 2.0.
Good morning to all. On behalf of Department of Biotechnology and organizing team of SBT 2.0, with the permission of dignitaries on the dais and convener of SBT 2.0, I, Dr. Ambika Arketkar, propose a vote of thanks. The symposia was proposed, planned and executed in the shortest time duration. When time frame is small, the support system has to be very cohesive. I take this opportunity to extend a heartfelt thank to Gujarat State Biotechnology Mission for financial support. The officials, Dr. Snehal Bakhtaria, Joint Director GSBTM, Shri Digvijay Singh Jadeja, Mission Director GSBTM, and Dr. Mitali Dabi took very prompt decision while planning SBT 2.0. Thank you, Mitali Madam, for your continuous support for both the versions of skilling biotech talent at the National Symposium. I humbly thank Mr. Rahul Kharadi, Deputy Manager GSBTM, for gracing the occasion and always supporting Department of Biotechnology for different programs conducted under GSBTM. Thank you, sir. Being a joint venture, if things move faster from one end, then it is must that they can move faster from the second end also. I take this opportunity to extend my gratitude towards Honorable Vice Chancellor Dr. Chavda sir and respected Registrar Dr. Gadvi sir from Veer Narmad South Gujarat University for their support and encouragement in organizing SBT 2.0. I would like to extend gratitude to Dr. Narendra Bhai Patel, in charge Registrar, for gracing this occasion. SBT 1 and SBT 2. Both the symposiums were in association with Internal Quality Assurance Cell, IQAC, PNSGU. I thank Dr. Desai sir, Director IQAC, for his willingness to join the program and support that he has always shown for SBT as a national symposium. The point of attraction in SBT is always its expert panel. I thank Dr. Bakulesh Khamar, Dr. Rajesh Patel, Shri Kamlesh Yadnik, Dr. Ramesh Kothari, Dr. Jay Bargi, Dr. Mehul Dave, Dr. Pinakin Dindokia, Dr. Vijay Kumar, Dr. Pithawala, Dr. Anish Sharma, Dr. D.K. Joshi, Mr. Kapil Shah, Dr. Jyotirmay Aichi, Dr. Vivek Dikshit, Dr. Jigar Rana, Dr. Meghna Advaryu, and Shri Mayank Sharma. Thank you all the dignitaries for accepting our invitation and joining SBT 2.0. As our convener has mentioned in his address, we have received massive representation or registrations for SBT 2.0 from different sectors of science fraternity. I congratulate and thank all the participants, respected faculties and students for being a part of this symposia. Please be an active participant in panel discussion as well. Organizing an event in a short time span is the toughest part. Thus, I extend thanks to all the teaching and non-teaching members of Biotech family for their support. A special mention goes to Dr. Nikunj Tandel and Dr. Chirag Prajapati, organizing secretary and technical expert for SBT 2.0, who are working reluctantly for the success of this program. Thank you, Jagdish Bhai, for pouring your blood and sweat. I think he is outside. For organizing this event. Thank you, Ridhima, who is and who will be hosting this event. I take this opportunity to appreciate the efforts of students, whom you have already seen here. Student from semester 10 Jainil, MSc Biotechnology program, and other students from semester 4 BSc Microbiology and semester 6 BSc Biotechnology students who designed our SBT information booklet under the guidance of Dr. Nikunj Tandel and will be associated with the event for the rest of the day. I express my gratitude towards Dr. Gaurav Shah, convener of SBT 2.0, for his dynamic leadership and understanding the need of such program in the science fraternity. Let us all join hands and do a fruitful churning of thoughts for betterment of skilling talent and new avenues in biotechnology. Thank you.
Thank you so much, Dr. Ambika. And now it's time for our national song to be played. Along with national song, then we'll proceed with national anthem. So let's all together make our mark.
श्यामलां मातरं वन्दे मातरं शुभ्रज्योत्स्ना पुलकितयामिनी फुल्लकुसुमिता द्रुमदलशोभिनी सुहासिनीं सुमधुरभाषिणीं सुखदां वरदां मातरं वन्दे मातरं कोटिकोटिकण्ठकलकलनिनाद कराले कोटि कोटि भुजै धृतखर करवाले अबला के नुमा ए तो बोले बहुबलधारिणी नमामि तारिणी रिपुदल वारिणी मातरं वन्दे मातरं तुमि वेद्या तुमि धर्म तुमि रिद्धि तुमि महिमा त्वं हि प्राणा शरीरे बाहुते तुमि मा शक्ति वन्दे मातरम्।
And now let's have the national anthem. So please keep standing.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जन गण
Thank you so much. Kindly take down your seats. And now, moving ahead, as we have completed our inaugural session, now it's time to start our technical session. Ma'am. Ladies and gentlemen, hereby we would like to begin our technical session. The first session is on translating laboratory research into market-ready therapeutics. The session will be taken by Dr. Bakulesh Khamar. Dr. Bakulesh Khamar is Executive Director R&D at Cadila Pharmaceutical Limited, India since 1996, and he is responsible for bringing innovations at Cadila. He has his professional experience with M&J Institute of Ophthalmology, Ahmedabad, NHL Municipal Medical College, Ahmedabad, and Shankara Nithyalaya, Chennai, and School of Optometry, Berkeley University of California, USA. One of the key focus area currently is use of immunotherapy in various diseases to improve outcome without significant side effect with focus on cancer and sepsis. He has 130 plus granted patents in India and abroad and has several other patent applications under various stages of prosecution in India and abroad. He has more than 70 publications in form of research papers, book chapters, etc. As he is going through a little bit of health issues, so he is unable to make his attendance right now, but he will be with us in the session through virtual mode. So let us all put hands together for Dr. Bakulesh Khamar as he is the first to start this wonderful session. Let's hear a round of applause, please. Thank you.
For the invitation, and I apologize to all of you. Though I promised to remain physically present, I am unable to do so because of the health problems which happened recently. So, are we ready with the slides?
Sir, shall I move the slide?
Yeah.
Okay.
So, the next slide.
Next slide. Okay, sir.
So, the laboratory research generally generates new knowledge and therapeutic development of that knowledge is a complex multi-step process. Knowledge related to target, pathways, mechanism of action or a molecule can be considered for translating into the therapeutic products through a multi-step process. Multi-step process for developing therapeutics is broadly divided into two broad phases. discovery phase and development phase. Discovery phase identifies lead compound for development of therapeutics through animal and human studies. Discovery phase generally involves lab research, while drug development consume around 12 to 15 years and has a high failure rate. As shown in this slide, even when you have some lab research which is translatable, 31% does not make it to the preclinical studies. And for each subsequent stage of a clinical studies, the failure rate is maximum at phase 2. If you cross phase 2, your chance of having a successful therapeutic goes high. Next slide.
Yes, sir.
Discovery phase is for translating knowledge into a compound for further development. It involves laboratory research and academic freedom is best suited for it. Further development is better done by industry. Next slide. Design and mandate of academic institutes worldwide allows them to have a domain experts with better freedom for knowledge creation. Drug development involves interdisciplinary teams working and generally available under one roof in industry and not possible in academic institutes. The freedom which is enjoyed by academic institute does not exist in the industry. I will try to provide you bird's eye view of process of translating laboratory research into therapeutic products by two examples. One, when the regulations were not too strict, and one current examples. As you know, Nobel Prize in Physiology and Medicine is awarded for outstanding lab research which has benefited mankind. I have selected discovery of penicillin for elaborating on process of translating knowledge to the therapeutic product before regulations became stringent. Nobel Prize for discovery of penicillin was awarded to 3 persons, Fleming, Florey and Chain. Next slide.
Yes, sir.
As you see on the red line box on the extreme right, in 1928, penicillin was first observed by Fleming in the lab. But if you trace back the pre-existing knowledge, ancient knowledge taught that molds can be used to treat infections. And then if you look at all the boxes, they are divided into two colored boxes, two types of colors. One type says that penicillium, which was responsible for the penicillin, was well-known mold, and it was also known that it is capable to have antibacterial substances which inhibit the organisms. And other colored boxes suggest that one organism inhibiting growth of another organism was also well known. So this was all well known. So what made a difference for a Fleming? So it takes us back to the 1928 to 29, when Fleming was working at St. Mary's Hospital, London. He was a bacteriologist, and his research involved, at that point in time, he was trying to look at what are the variability in growth pattern of Staphylococcus, which is a pathogen. His prior research before this involved around lysosome and its lytic properties. Contamination of culture by mold was well known, and it was a common laboratory practice, at least at that point in time, and I believe even today in the bacteriology labs, to discard such cultures as soon as possible. However, as per serendipity, it was not discarded for 6 weeks, largely because he went on a vacation between that period. What made it special was his subsequent steps. He observed, next step, he observed contaminated culture to have no growth of staphylococcus around the mold. His prior work on bacteriolysis due by lysozyme and knowledge of bacteriology excited his curiosity. And because he became a little curious, he did something which nobody before him did. He isolated mold and conducted experiments like any good scientist to reproduce the results. After he could reproduce it, he went on to develop methods to isolate antibacterial substance from the mold culture and evaluated it further for its nature and solubility. He coined the word penicillin for the first time for this newly identified antibacterial substance from a mold culture. Next slide. He found penicillin kills some gram-positive organisms and not all the organisms. But all these gram-positive organisms were pathogens. What surprised him most was its potency. At that point in time, antiseptics were largely used in laboratories as well as in the hospitals. Penicillin was effective when it was diluted 800 times. This high potency was not seen with antiseptics. Another point which he noticed was, after experiments, that there was no effect on leukocytes of penicillin. Antiseptics are known to inhibit leukocytes or destroy leukocytes. And this differentiated penicillin from prevalent antiseptics. And subsequently, he also did preliminary safety studies in the rabbit and mice and found it to be safe. And with the help of his friends, he tried to isolate what the mold, what it is, and he found out that it is a penicillium rubrum. However, he could not go beyond to isolate and purify a substance, and this can be attributed to inability to recognize collaboration with a biochemist. Next slide.
Yes, sir.
He provided molds to the other researchers before stopping working on penicillin in 1929, and he hoped that other scientists will continue work and find out something. But he made use of this penicillin only to isolate and purify active substances. Separate the organism. After he stopped working, his work significant was continued to have something different, but he could not think of that it can be used to treat infection, and he continued using it for isolation of one bacteria from other. And after publishing that, he did not continue this. In 1932, Reistig, the scientist with whom he had shared the culture, was a biochemist at the London School of Hygiene and Tropical Medicine. He got interested and he reproduced, as a first step, he reproduced Pellin's results. And being a biochemist, he designed a synthetic media to grow the mold, and he was also able to isolate penicillin in an active form and also develop assay for efficacy of penicillin. Till this point, penicillin is a crude extract, it's not what we know today. He also identified conditions to store it for up to 3 months. The penicillin was highly labile compound till that point. Next slide.
Yes, sir.
Prior approval of sulfonamide in 1935 as a chemotherapeutic agent to treat bacterial infection, there was notion, defied the notion working at that point that chemotherapeutic agents will be too toxic to use for treatment of infection. So that was a significant change in the mindset of people that chemotherapeutics can only be used for antiseptic, but this was the first compound which suggested that it can be used for a systemic infections. And this led to initiation of research to develop antibacterial agent. In this background comes the next level. So Fleming also transferred this to Oxford University, Sir William Doon School of Pathology, which was established in 1927 with a specific purpose to move the science forward. Fleming had sent their culture in 1930. The director was working on antibacterial properties, mainly through the bacteriophages. And he thought this can be one of the bacteriophages. And when he realized this is not a bacteriophage, he stopped working on penicillin. But scientists in his lab continued working on it even after he died in 1934. And she continued using it to isolate one bacteria from another, specifically for his work on influenza. Next slide.
Yes, sir.
In 1935, Florey became the next director. The Florey, as you know, was a recipient of Nobel Prize for penicillin in 1945. So 1935, he becomes the new director. And he invites Chain, who was a co-participant in the Nobel Prize, to join this institute. And initial projects they worked on was lysosome and snack venom, primarily to identify natural products with a biological activities and try to understand how it happens. So, next slide.
Yes, sir.
When they were on the close of completing this project, as each lab will do, they started looking for the new projects. And during that time frame, Chain got interested in the literature around inhibited substances produced by one microorganism to inhibit the growth of another one. And then he went into the greater details, and extensive review of literature and discussion with Florey led to focusing research of that lab, and which they thought this will be a long-term project related to chemical and biological nature of some inhibited substances, with the hypothesis that a specific enzyme will be responsible for this antibacterial activity, which can be isolated and synthesized. And for initial project work, when they wrote a research project, they included 3 microorganisms: Bacillus subtilis, Turpella pyosinus, and Penicillium, which was identified by the Fleming. Next. After receiving the project approval, the next thing which Florey did, and which differentiated him from the others, he created a multidisciplinary team at a Dunn school, and as shown in this slide, with a specific responsibility related to their expertise. So this team had a biologist, biochemist, clinicians, technicians, all kind of people put together, and this was a 9-people team with Florey and Chain at the helm of efforts. Next.
Yes.
In 1939, they started their work on penicillin, and as a first step, they confirmed the finding, and they were satisfied that, okay, penicillin is a good candidate to work on. And they focused on penicillin, discarding the other 2. In 1940, contrary to their initial hypothesis, they found that penicillin is a low molecular substance and is not an enzyme. So an average researcher would have lost interest because you are trying to do something which is not there. However, they pursued the work, and developed methods of purification of penicillin, and they improved to obtain penicillin in a solid form. In August 1940, they conducted first animal studies, and this was for the first time anybody did. Before that, everybody did in vitro studies or culture studies, but they were the first one to do with any antibacterial substance of a origin of the fungus to evaluate its efficacy in penicillin in a mice model of a lethal streptococcal infection. And this was found to be highly effective, and the animals or mice which received penicillin got cured, while animals which in the control arm, all of them died. And they published this in a Lancet, which is, if you know, is highly reputed journal. It generally publishes articles of clinical relevance or clinical trials, but they published this article as an exception. Oxford team continued working and published their findings related to growth characteristics, and which was published again in Lancet in 1941. Next slide. In 1941, they could have some amount of penicillin as a barium salt, which was of a significantly pure form, and they standardized the production of penicillin, establishing efficacy and safety in animal studies. So original studies were done in 10, but then subsequently did more studies involving 80 animals each time and multiple times. And they thought that now it's time to start clinical evaluation. And then, as you can see, there was a signs of improvement, but none of the patient got cured, as the treatment cannot be continued due to lack of adequate supply. So when they started giving it, and this was done without knowledge about amount of the drug to be given, the frequency of the drug to be given, but they still observed recovery for the early periods till they could continue giving penicillin. And once the penicillin they could not give because of the exhaustion, everybody succumbed to the infection. And so they said that now this is going beyond a lab research, and they tried to approach the industry in Britain. However, the response was not favorable. So in 1941, Florey traveled to US with the help of Rockefeller Foundation to explore opportunity for large-scale production and further clinical evaluation. He first visited US Department of Agriculture, North Regional Research Laboratory, Illinois. Why selected this? Because this lab was pioneer in a large-scale culture using a submerged culture. Till that point, everybody using a surface culture. Surface culture are not known to produce large amount of the substances. So they were pioneers. So he went there, they had a discussion, and the lab scientists suggested that there are a lot of things which can be done. Some of it is culture medium can be altered, and you can also look for a isolation of high-yielding penicillium strains. After that, they agreed to work together. Florey next visited Richard, who was chairman of a committee of medical research. This was created in 1941 by President due to the war condition for meeting the military requirement. So Richard, looking at everything, got interested, and he promptly organized teams by convincing Merck, Squibb, Pfizer, Abbott, and launched a project, synthesis of penicillin, to make penicillin available for clinical evaluation. While this was happening, the lab which Florey visited first in Illinois, NRRL, got their data out, and they suggested that it's possible to have a higher yield of penicillin. And recognizing all this, UK and US got together and decided to work together as a one team to accelerate development of penicillin. Next slide.
Yes, sir.
With this use of American resources, very quickly penicillin became available in larger quantity in US compared to UK, and US started using it after confirming the animal data and safety. They started using it in humans. So first human to receive penicillin in US was in 1942, and the lady was successfully treated with a penicillin. So that was the first success story of penicillin in human. Following that, there was a lot of small studies, but Army got interested, and first large-scale study was done in 1943 in a Brigham General Hospital in Utah. They had 200 patients with a complicated fractures, complicated by osteomyelitis, and as per the standard treatment at that point in time, all of these were supposed to die because sulfonamide does not work here. So they immediately started using penicillin, and they applied it on the wound, as well as they went to give it through various parenteral routes like IV or intramuscular or subcutaneous. And they got highly encouraging results, which were shared with the Army headquarters, and immediately the same experiment was repeated in another hospital in New York with again 200 people, and having noted safety and efficacy, Army went on large-scale use of penicillin in the wartime, and they trained large number of people in these two hospitals and sent them around the world, and penicillin produced in USA was tightly controlled for Army use, barring few civilian use. So at that point in time, only 20% was available for research and civilian use. Next.
Yes, sir.
And this is the first publication for efficacy of human, which was not successful, but it was the initial data from Oxford team. Subsequently, UK and US team, industry as well as academia started working on the various aspects of product development. There are large data and reports were generated which were classified and not made public. Only some parts was allowed to be made public. And I'm just showing you a few of the examples of what was done. So Oxford team and another Imperial College team in UK standardized the purification system for a penicillin. Sterility and stability was done by various pharmaceutical industry in USA. Assay which is necessary for release of the product was initially developed by industry in USA, UK and Canada. The formulation need to be stable, and it should be, reasonable time, and this work was again done by the Army Hospital in UK, and for any product to develop, before it goes into human and you standardize it, you need to know absorption, accretion and distribution. This was actually done in humans and not in animals, unlike the current time frame. And they were done on patients who were being treated, and this was done at both the places, US and UK. And as you see, Johnson & Johnson supported the research in US. Then you need to know what amount of drug is available at what time frame, and this is important for when to repeat the dose and how much amount to be given. And this was done in the patients with infections of various types, including gonorrhea. And then they showed that when you do it in the serum penicillin levels falls very rapidly. It comes to 0 by the end of a second hour. So if you don't repeat the injection, patient will not be cured. And 60% of it is excreted in urine in 1 hour. So, and these were probably the reasons for failure of the early clinical trials done in UK. Team in Bachelor's lab worked on the gonorrhea, and then they standardized the dose based on the clinical activity. And they suggested that a single intramuscular dose is effective in very 20%, but if you give 2 doses 6 hours apart, it's effective in 55%. And then subsequently, the dose was increased, and gonorrhea being highly specific, they could control the gonorrhea across the army. Studies to determine were again done in Boston. So various parameters. So when you give a drug, you need to know what parameters will affect the availability of drug for its efficacy. And this was done in Boston. And then people started using a larger number, and publications started coming in a large number. So this was the first largest study published, 500 patients in JAMA for Journal of American Medical History. And then in army at that point in time, sexually transmitted disease was a major problem. And the studies were conducted by army in syphilis and gonorrhea, and they got published. So I will stop here the story of a penicillin and take it to the next story, development of a rabies vaccine, which is our own story, and it has gone through the stricter regulatory environment of the current times. Next slide.
Yes, sir.
Now, before we want to there, I mean, you need to look at how the rabies vaccine evolved over time. So when Pasteur identified rabies vaccine and developed it, number of injections were not standardized. It was 18 or more. Over a time, as they started purifying the product, it became 14, as many of you may know, because it was grown in the nervous tissue, and subsequent science and technology development improved, changing it from a brain or a spinal cord to the varocell or cell-based or egg-based media, and improved purification, reduction in impurity, reduce the number of rabies vaccine from 14 to 5. However, in the Indian scenario and around the world, majority of people will take only 3 injections because of the complicated regime, because it has to be taken day 14 and day 28, which is not in alignment with day 1, 3, and 7. So this was a major cause of failure of a rabies vaccine. And we thought that can we do something, because it's a problem of a country like ours. No Western world or a developed country will look at rabies, because large number of deaths takes place in India and other developing countries. As a matter of fact, 22,000 people, which is highest in the world, dies of rabies in India. So we looked at what are the options available. So essentially, you have to improve the immunogenicity. Originally, it was done by removing the impurity or improving purity of the vaccine. So what are the other mechanisms? You can use adjuvant. So, I mean, people have tried using various adjuvant. One of the known is alum, but there are other adjuvants. And they were not satisfactory. They were not able to reduce the number of injections. At that point in time, we were running our own adjuvant program. And our adjuvants, when evaluated for a conventional rabies vaccine, we could get to the 3 dose, but not everybody get protective titers on day 14 in the humans. So in animals, it was good, but in humans, it was not good. So that was not a good idea. So then the next thing is, you have to look at how do we improve the antigen profile. Next slide.
Yes, sir.
So the science, lab science, suggested that the antigen for a rabies vaccine is a rabies G protein. It's a very small amount, as shown in a red circle in the figure. So it contains a very tiny portion of the whole virus. but we are using whole virus. So we thought that if we can produce this protein in a recombinant way, probably we might have an answer. But the efforts made to have a G protein isolated and use it as a vaccine were not successful because of the stability problem. Next slide. But using our technology, we were able to produce rabies G protein and make it in a stable form. And that started the journey of a development part of the rabies vaccine. Early preclinical studies, I'm just showing few slides, but there are lots of studies done, suggested that it's significantly better in immune response compared to the conventional vaccine. So as you see in this slide, we compare our product with a Rabipur, which is a standard inactivated virus vaccine, and you see that it provides immune response which is earlier and higher and sustained compared to Rabipur. And then you need to characterize the product well, which includes physicochemical characteristics, biological characteristics, its potency using the standards, expression profile, impurity profile, toxicity. So completing all preclinical studies, we embark on a phase 1 study. Now, we never knew, because nobody has done this before, so we designed a phase 1 study which had 17 arms. Generally, phase 1 studies are generally 1 arm or at the most 4 arms. But this study was unique. It had 17 arms, and we evaluated various regimens which can be divided into 2-dose regimen or a 3-dose regimen with an amount of rabies G protein 5, 10, 20, and 50. When the results became available, it was found to be effective, and we did a phase 2 study and selected based on the data, and this was again a 5-arm study. So 10 and 50 microgram, 2-dose versus 3-dose. So that was the next level, and findings suggested that the best results are obtained with a 50 microgram 3-dose. It provided us an adequate margin of safety. Though 50 microgram 2-dose was equally effective, we considered 3-dose considering adequate margin of safety. And we did a large-scale phase 3 study. This is the largest study ever done for a rabies vaccine. It had 800 volunteers, randomized in 2-to-1 ratio. There were 535 volunteers in the test arm receiving 3 injections containing 50 microgram of rabies G protein on day 037. Control arm had 267 volunteers receiving 5 injections of inactivated virus vaccine, which is standard of care, and they received a full course, which is 0, 3, 7, 14, and 28 days. So 5 injections versus 3 injections, recombinant protein versus inactivated virus. And when the data became available, as expected, safety was significantly better with a recombinant virus protein vaccine compared to the inactivated virus vaccine, because it had a less impurity. And efficacy-wise, it was found to be identical, and that led to approval of the product. So essentially, when you are thinking of translating laboratory research to market-ready therapeutics, you need to have an interdisciplinary team with expertise in science, technology, and regulatory aspects which are required at various stages of development. Thank you very much.
Thank you very much, sir, for your presentation and explaining penicillin, the journey of penicillin, as well as how rabies vaccine has been translated from laboratory to market-ready therapeutics. I am really obliged to have you in virtual mode for this one-day symposium. Thank you very much, sir, for your presentation. Thank you.
Thank you so much. And now let's move ahead with the next session, which is on emerging research funding opportunities, national and international landscape. And for this, the session will be taken by Dr. Rajesh Patel. He is working as a professor in the Department of Bioscience, VNSGU, Surat, India. He is the coordinator for NIRF, UGC, SAF, and DRS second project. He was awarded as a young scientist computer facility Param Sevak at VNSGU Surat, and he was awarded as the Young Scientist Travel Fellowship from FAOBMB Center for International Scientific Exchange, Chinese Academy of Space, Beijing, China in 2000. He was also awarded a fellowship from UNESCO and ICRO for the International Training Course on RNA and Biotechnology, State Key Laboratory of Molecular Biology, Institute of Biochemistry and Cell Biology, Shanghai, China in 2000. His current research areas include extremophilus, microbial enzymology, and metagenomics and drug discovery. Dr. Patel has published 72 research papers with 1282 citation and also contributed to 9 books. Dr. Patel utilizes his educational and research capabilities and ability to impart knowledge effectively to assist students. So I request Dr. Rajesh Patel to please come forward and take the podium.
First of all, I would like to thank the Gujarat State Biotech Mission and Department of Biotechnology, who has given me the opportunity to have the interaction with you all. So the topic assigned to me is about the emerging research opportunities at the national and global landscape. So as the basic theme is about the skilling biotechnology talent, and it is about the skilling the biotechnology for the next generations. So my presentation would be in the 3-4 different parts only. We'll be taking some of the example and how you can develop your career path in the future. So we are starting with the current research progress. So suppose you are finding the entire world, then you can find out that research is the engine of the modern progress. Now the GDP is calculated in terms of the R&D proportion, that how much GDP is developed, that would be proportional to the R&D investment in the country. The universities are not only a institute where you can just have a degrees, but that is now becoming slowly an innovation hub. And in this era only, the talent should be a strategic asset for any university. So in that context, the entire symposium which is developed today, that would be a nice step for developing the entire university in an innovation hub in the future. Now suppose you are finding the funding pipeline innovation in the world, then you can find out that suppose we are starting from the different countries, then you can see that we are just lying very behind from all of this. India is coming around 0.5 only. So suppose you are comparing the Indian biotechnology strategy, then that is also around 1.50 billion bioeconomy, but still the government investment is quite less. So that requires the more amount of the investment in this. The basic translation challenge is quite huge. So we have seen the example before this only, that how the penicillin was converted or how the rabies vaccine was developed. So that includes the entire path. So that will always start with the basic research. Somewhere in some lab, somebody is finding some basic principle. After that, by doing some lab work experience, they are just converting them into the applied research. Suppose you are taking the example of the penicillin, then earlier that was developed in the tubes only, and after that, that was developed in the bottle. And during the World War, that was also produced from that only. But slowly after that, when it is coming to the industry, the technology is getting transferred. The industry would be producing at a very high amount. And from that only, the market impact would be created. The similar thing we also experienced during the COVID also. So initially, the things were developed in the lab, and very rapidly that is converted into the market. So that is a translation challenge, that whatever is there in the basic research, how it can be converted into the translation research, and how universities can convert their research into the innovation. So for that, you can just have the entire map like this. So now university is having the growing role. So university will remain in a center, but they should have some kind of the government agencies like the GSBTM and the will be funding for the entire part. They should have some kind of the global research network so they can find out that what are the trends are going in the world, and from that only they can just create some kind of the funding project, and from that funding project only they can start something which is useful for the society and end product. So to develop that only, now all the industries and all the universities are collaboratively producing the different kind of the startups and incubator. So the product would be getting incubated and startup prototype would be developed, and if it is getting succeeded, then it will go into the industry partner. So that way, suppose you're observing the entire role of the university, then it will be just starting from the seeding project from the academic part, and that is going as a huge tree in the field of the industry. But that requires a lot of fund and lot of things from the government agencies and other. Now this global transformation of the research ecosystems are there, and now academic is also converting into the innovation part, so that already we have seen that India is lagging behind in this. Now suppose you are taking that what is the leading innovation race throughout the world, then you can find out that United States that is coming first, but they are majorly investing in the AI, deep tech technologies and other. So they are the top universities, they are creating the different kind of the AI platform and AI tools only. Suppose you are going to the China, then you can see that the China is going for the quantum computing, they are working on the 5G, and they're also going for the manufacturing process. You can see that almost each and everything we are consuming, almost more than 50% is coming from the China. So their focus is on the manufacturing. Suppose you are going to the European countries, then you can find out they are working with the collaboration of the research, but their focus is on the sustainability, on all the health also. And suppose you are going to the Korea, then you can also find out they are working on the electronics and other. So each country will have the different kind of the priority, and on that area only normally the government is funding, and the research is converted in the innovation part, and even it is getting commercialized. So suppose you are taking the India in this race, then you can find out that we are having the mixture of all of this together. We are not having the one tag on which we are having our clear policy that will working in this area only, and that is one way good for country big like us also, because we are a huge country and we are having the lot of potential. So instead of focusing on one, we can work on the many frontiers together. So currently the technologies which are growing in the market, that is of course the artificial intelligence, biotechnology, robotics and quantum computing. So I'll be taking some of the example that is normally required for the research. Suppose you are taking this, then you have to also take that what are the global grand challenges demanding the research solutions. So as I told earlier that the research cannot be done in the silos, the research should be done for the society. And suppose you are taking the grand challenges in the world, then we are having the global challenge of the climate and energy. Right now we are facing the problem of the LPG because we are depending upon the Gulf area for that. Similar to that, something is happening in the climate, then we are totally helpless. So the biggest change for the entire human mankind is the climate and energy. The second part is the global health. So we are getting the lot of communicable and non-communicable diseases. Of course, we had the era of the COVID and we had the lot of experience from that. But besides that also there are not many non-communicable diseases like the heart attack and other that is also getting rise. And the future pandemic would be again of the antimicrobial resistance in 2050. So that is again become a global health challenge for us. Similar to that, we are also having the problem with the food security. So our population is getting increased, and due to that only, suppose you are comparing the land which is required for that, that is getting shrink. So there is a huge gap between the food production and the food consumption. So that becomes also a main part for the global challenges. So for solving all this problem, what you require is you require the interdisciplinary research solutions. So you require the biology, computer science, engineering, social science, material science, and this everything is coming in the form of the interdisciplinary science. And the academia industry collaboration, so the workshop or the symposium like today, that should have the basic research so they can find out the explore the fundamental principles, and after that only they have to just go for the developing the prototype, and that can be transferred in the technology format, and from that only the commercial product can be converted. So the university can become the entire path from that, and research will become the national competitive advantage for that. So this is the global innovation ranking where India is trending. So you can see that the India is coming good in position with the comparison of the lower middle economic company that is in the 13. But suppose you're comparing the entire world, then India is ranking around 40th. So that way still we are having the lot part to cover by us. So the future of the scientific research, so that is starting with the open science. So we have to go for the publishing the data in open access journal, and we should use the data, and from that only open science concept only the research can be accelerated accelerated. You should have the AI augmented research. Your capability of doing the research can be increased to that. And we have to focus on the citizen science. Means whatever you are doing, that should be some way or other that is helpful to the some citizen of the global or at the national level. And your entire science should be the mission drive session, right? So these are the few things which is normally coming. And translation research into the innovation is the many part. When we are coming to the normal research, then we are having the one experience that is known as the valley of death. The valley of death is funding gap between the early stage research and the commercial viability of the claim. You can see that in each university we are having the hundreds of the students who are doing the PhD. They are publishing so many papers, but out of that only few papers are coming in a commercial form, or the few ideas are getting commercialized. So this is basically known as the valley of death. So whatever the research is there, that is just get dumped in this part. So there are misleading kind of the incentives. So academic reward structures are prioritized basically upon the publication and citation, not the market outcome. Suppose you are taking the current guideline of the UGC for the minimum appointment of the assistant professor, then also you can find out there is the points for the publication, there are points for the projects, there are points for the patent, but there are no point for the research outcome in terms of the some kind of the product directly, right? So this is diverting our entire youth in such direction where public are just going for the citation, publication and everything only. The translation barriers are also there because in country like India, we are having the huge regulatory constraints and we have to pass through even all these constraints. Even in our university also, whatever the regulation committee is required for the research, still some reason is that we are not able to maintain it properly. And suppose you are going for the any research in the biotechnology, then you can have also the lot of regulatory research and other. So this is a basic gap between the academia and industry. So academia is basically working for the knowledge generation, and that is the long time horizon. Publication, public funding and open sharing. That is a task which can be done by the universities. At the same time, suppose you are going to the industries, then they will go for the market application, short-term time horizon, they will just go for the revenue matrix, that will go for the private investment and they will propriety or they will protect their IP protect another. So as the academic is having the open part, where the industries are having the very closed system. And that is a dilemma in the Indian industry that the industries are not coming in the university and universities are not thoroughly going to the industry. So that way we should develop some kind of the mechanism by which at least some kind of the sharing is possible between both of this. So that is basically a part of that. The technology readiness level, now suppose you are taking the different project, then you can see that even when you are going for the applying the project, even in the funding agency like the Gujarat Biotech Mission, they are also asking that what kind of the technology readiness level you are doing. So basically you can have the technology readiness level that is starting from 1 to 9. So whatever the normal research is there, that is residing into the 1 to 3. So that is concept of the proof. And normally our 95% or even in some case 98% of the research are rotating around the basic research only. And due to that only commercialization of that is not possible. Applied research is a research area where we are just starting from the TRL 4 and that is going to the 6. So the lab would be producing some kind of the prototype that can be taken up by some industries and that can be commercialized in further. At least if it is getting failed, they will get the idea that okay, these things are not working. So that kind of the projects are normally residing in the 4 to 6, and after that you can have the commercialization project that is coming in the TRL level 7 to 9, and that is from the system to the development. So now the universities have to align their research work that should not go in the only area of the 1 to 3, but at least they should start with the 4 to 6. So as a researcher, when you are going in the future, when you are writing your thesis or when you are writing any project, you have to take care that whatever your research is there, that is crossing at least the 4 and above boundary for the applied research. So the university will become innovation ecosystem, but for that you should require the research lab, technology transfer office, incubators, accelerators, and you require also the industry partners. So some of the things are very common. We all know about the incubators, but along with the incubator, there is one more word that is known as the accelerator. So accelerator is the area where you can just do the science, you can do the product development, but that would be done in a very accelerated form. You're given a mission-like object, so you'll be given 3 months or 4 months. In that 4 months only, you have to complete your entire task. So now your incubator should be converted into the accelerator. And after that, as per the NEP also, we should develop the technology transfer office, and that can be utilized with the industry partner for the further development. So these are the role of the incubator and accelerator. So along with that, suppose you are not able to go for the commercialization of the any product, then what you can do at this stage of the university is you can just go for the filing the IP. So you can just go for the patent, licensing, trade secret, copyrights, trademarks, or you can have the material transfer agreement. So by this, at least your protection of your rights would be done. So you can just have the very common example that right now we are using the Coca-Cola tin, no? So that Coca-Cola tin is having the one kind of the design by which you can open that Coca-Cola, right? So that design was invented by one person. At that time, the people were not having any kind of the utility of that time, because at that time everybody was moving with the knife in their pocket. So they can open the bottle with the knife only. So he thought that this invention is not of any use commercially at that part only, but he has created the patent of that. But after that, when the real Coca-Cola tin came and the people were not having the knife, suppose you in this room you want to open the Coca-Cola tin, then you can utilize that design. And for that design only he has taken the revenue. So that is the way that whatever the things today it is not getting commercialized, but if it is a good idea and if you are protecting with the IP, then in future you can have the entire thing from that only. Similar to that, for the Coca-Cola also you can have the trade secret. The formula of the Coca-Cola is not known to anybody. Even whatever the ingredients are there, that is stored by the 2-3 different person in the world, and they're just mixing the entire thing. That shows the power of the entire thing. In Surat also we are having the very well-known cold drink brand, no? We are having the Socio. So you can see that we had the Parle, we had the Coca-Cola, we had the so many other brands, but still Socio has maintained their trade secret and they remain as such only. So that way you can also maintain your intellectual property right and other. So now what you have to do is you have to convert yourself from the researcher to the founder, and that journey is starting like this only. So you should go for the university industry, collaboration, meet and other. So these are the few case studies where the biotech companies are converted from the lab to the entire commercial part. So you can just have the example of the MIT. So they've gone for the creating this some kind of the vaccine in the lab, and after that only that lab vaccine was getting licensed, and from that only the huge Moderna entire concept has came. Similar to that, even from this CRISPR-Cas9 genome editing, some initial part was done in the Editas Medic, and after that only that is commercialized. Oxford is a very well known in this. Even in the Oxford lab also a lot of things are done, and even today also we are using the Oxford nanopore sequencing. So that is also came from the research lab only. So that is a way by which you can just have the conversion of that part, right? So this required the entire innovation culture, and to build this innovation culture, what we require is we require the awareness. And I think today we are at the first stage that we are just creating the awareness. After that we should go for the engagement, then we should have the infrastructure. Just near to us only we are having the new building which is coming up as a research center. So that can be a part of the infrastructure, and then you can have the integration, and finally you can create the ecosystem in only the work can be done. So suppose typically we are taking the overview of the Indian research ecosystem, then you can see that as per the NEP 2020, we are focusing on the multidisciplinary research. We are going for the internationalization. We are also promoting the industry academia bridge, and we are also talking about the equity and access. So our paths are set in a proper way, and if you are going further, then you can just have the more benefit in this. And looking to this only in the India, now all the funding agency, most of them, they are clubbed together and they have created the one common kind of the platform that is known as the Anusandhan National Research Foundation. In short, that is also known as the ANRF, and that is having the funding of around 50,000 crores. So like that only you can see that that is including all the schemes together only, and from that only you can have the different part. So basically ANRF, that becomes apex body, and that is including the different bodies like the DBT, DST, ICMR, CSR, and even they are having the lot of institutional part like the IIT, IISc, AIMS, universities and industry. So whatever the funding agencies are there, that is also getting changed, and now the Indian ecosystem is also getting increased. So now we are having the DP IIT recognized startup of around 1.4 lakhs. So these are the startup which is already in the market. From that, more than 110 big startup that is normally converted into the unicorn that is developed. India is also having the 9,000 plus different kind of the incubators and accelerator, and even we are having the funding of around 10,000 crores in this area only. So now the role of university is also getting changed in this only, right? So that are the few things about the internal part only. These are the few DBT flagship research project which is normally required. So normally now the government is focusing on the different aspect only. In that only there is one scheme which is known as the BioNest scheme. So this BioNest scheme is creating the bio incubator in a industry, or even it is also setting up the entire thing in the universities also. So BioNest is created in the university where you can just do the biotechnology related part. We are also having the genome Genome India project. So that is going for the sequencing of more than 10,000 different kind of the human genome, which is coming from the Indian territory only. So that will open up the lot of opportunities in the medicinal and drug development part. We're having the National Agri Biotech Mission, so that is focusing on the agriculture development. And again, we are also having the BioE3. We'll talk something more about the BioE3 later part. So this is the way the innovation mission of the India is working. So you can see that is started from the 2015, where only the startup were there. In the 2018, we had the Atal Innovation Mission, and in 2020, after getting the NEP, in the 23, now we are having the ANRF, and now strongly we are having the 2024 that is focusing on the BioE3 policy and other, right? So these are the translating the part of the entire part. And India's vision for the 2047 is increase in the GDP, top 10 global research nation would be there, and there should be the more than 300 billion of the bioeconomy, and again there should be net zero innovation should be done. Along with that also we are having the development in the industrial sector also. So now the industry is also getting converted into the different version. Suppose we are starting with the industry, then initially we had the industry 1.0. From the industry 1.0, what was happening? Normally we are just cutting the forest, we are cultivating the lands which as per our need, we are controlling the animal. That was the industry 1.0. Slowly we had the industry 2.0, where we have just developed a lot of things at a large scale. So that was the era where the different kind of the steam engines and others are created. And after that in the industry 3.0, slowly the mass production or the scale up of the industry was done. And in the industry 4.0, slowly we had the condition where we are just having the cyber physical part, we are having the IoT, big data, AI automation, cloud, and everything is came. So whatever the product you are observing right now, that is mainly of industry 4.0. But now in future when you will be going in the market, you can see that whatever the 4.0 is there, that is also having the lot of drawback. So that is having the human displacement, that is having the environmental cost, and that is also the fragile for the supply chain and other. And that is not the neutral value design. And that has given the more urge to the entire emergence of the industry 5.0. So when we'll be going in the market, you'll be finding that there would be an era of the industry 5.0. And in the industry 5.0, what you require is, you require the human-centric technology development, you require the technology which is, should be sustainable, and you should have the resilient kind of the design for each and every product. So that would be the era in which industry 5.0 would be working. So what is the human-centric technology? So earlier we talked about the robotics, but now we are realizing that not only robotic will work, but you should also go for the human intervention along with that. And the robot should not work only for the product, but the robot should work for the human goal also. So that is the area in which it is working. Again, we are just going for the sustainable manufacturing. So we are just going for the circular part of the thing. So we are just targeting the circular material flow. We are just increasing the bio-based inputs, carbon neutral processes, water stewardship, life cycle design, and again the renewable energy. Because we are knowing that Earth is a closed system where all the resources are limited. And if you are not maintaining these resources, then in future you will have the problem. So we require the sustainable product for that. Along with that, we are also having the AI development as a part of the industry 4.0, and again AI is developing a lot of in this case only. So AI and biotechnology is now changing the entire shape. We are having the AI driven drug discovery process. We are having the genomic intelligence, because due to this AI and even other kind of the quantum computing, it is very easy to go for the large scale data analysis of the genome. We are going for the autonomous bioprocessing. So bioprocessing is good, but the bioprocessing is not optimized by using the AI techniques and other, but now people are going for optimizing the bioprocesses. Similar to that, now even the era of the synthetic biology design has came. So instead of working on the living system, now we are in an era where we can just synthesize some of the mini creature, and from that we can just develop our product. So that are the smart biofactories. So that will include the bioprocess as a core, that will have the sensor and data, and that will also have the AI based part and other. Along with that only, the developing area is also taking the part where we are just going for the personalized medicine. You can see that earlier the medicine was common and that is given to all, but now we are knowing that the one medicine will not work with everybody. So you should go for the personalized medicine. So you should go for the patient genomic profiling. Currently also, suppose somebody is suffering from the cancer in your family, then you can always observe that after the biopsy, the tumor was sent to the analysis and the tumor marker would be detected, and depending upon the tumor marker only, the drug would be given to you. you. So that is also the one kind of the explanation of the personalized medicine. That is not the 100% personalized, but at least they will check that which kind of the medicine can work better with you. Similar to that, you can also have the on-demand bioprocessing, because you know that bioprocessing for a typical molecules which is consumed at a very low part, that is quite costly in nature. So in future you can have the on-demand bioprocessing. Suppose you are in need of some medicine, for you only that medicine would be created and that would be parcel to you. Similar to that, you can also have the adaptive supply chain. So that will be taking the logistic part and other, because many of the biotech product will require the cold supply chain and other. So this is the area in which it can work. Then you can also have the human machine collaboration. So you can see that full human control, that is the craftsmanship, but now we are having the human guided automationship, and now that is also going for the collaborative teaming, and you can have the cobot working for that, and fully AI system based human goal can be achieved by this. So now we are not having only the human machine collaboration, but we are also having the human machine and AI collaboration. So I'll be talking some more part about the circular bioeconomy. So circular bioeconomy is a bioprocess upcycling, that is closed loop system, and in which you can also have the ecosystem regeneration, and you can have the bio-based feedstock and other. So basically what we are targeting is reusing the entire waste which is generated into the some kind of the product. Suppose you are talking about the industry 5.0 in the agriculture, then what kind of the possibilities are there where the biotechnology can work? So you can have the precision biofarming in the future. Right now, suppose you are using the drip irrigation system, but this drip irrigation system is still only a mechanized one. You are just giving the water as per our need and as per our some time control mechanism. But suppose you are adding the special kind of the moisture sensor that will sense the moisture, and depending upon that only the water would be given, and that will be stopped as the targeted moisture is achieved. Then you can have a better part. Similar to that, the precision biofarming can be done for the pesticide, herbicides and other, but this require the AI guided sensor and lot of automation in that part only. Similar to that, we are also having the synthetic biology agriculture. So you can have the engineering microorganism which can fix, that can surpass the pathogens, and that can enhance the nutritional uptake and other. So currently we are in the era where we are using only the biofertilizers which are created from the natural resources, but we are not going for the much modification in this. So the future era would be of the synthetic biology in the field of the agriculture, and even some kind of the genetically modified crop would be also coming to this. Then you can also have the vertical control environment. So you can have the AI optimized farm control. You can produce the around the year with around 95% waste and zero pesticide and close to urban consumption centers. So that way you can also control the environment in a limited area, and you can have the higher production with that. The Bio E3 policy is also launched in the India, and as per the Bio E3 policy, that is talking about the economy, environment and employment. Earlier policies were talking only about either economy or they are talking about only the environment. But now we are realizing that if you are not engaging the employment along with that, the growth will not be sufficient. So this Bio E3 policy is basically talking about these three pillars together. So these are the few part of the Bio E3 policy, and this much grant is given, and mainly Bio E3 policy is targeting in the tier 2 and tier 3 cities, because you can see that industry 4.0, the cities like the Bangalore and Pune are getting well developed. Similar to that, suppose you are talking about the Surat, then in the Surat also we had the higher development of the textile, diamonds and other. But due to that only, there is lot of problem of the migration from the tier 2 and tier 3 city, and that is creating again lot of pollution and other problems in that. So this Bio E3 policy, it is targeting such kind of the solution in which the tier 2 and tier 3 cities can be developed as a manufacturing hub, and where the manufacturing process can lead to the economy, and that can also generate the employment, and with that way you can also preserve the environment also. So that is the basic target of this. So this is the vision of the bioeconomy. So bioeconomy target by 2030 is around 300 billion, and current value is only 150 billion. So that much increase is normally 2x increase is believed in that, and that will create the job target of more than 100 million. So now the target is not only the money, but the target is how much human manpower you can utilize in that part. So that is a vision of the bioeconomy system. For that you can go for the biomanufacturing ecosystem. So you have to develop the research. You have to go for the pilot scale up, and after that from the biomanufacturing you can go for the market deployment part. For this you can go for the different approach for the sustainable bioeconomy only, and in this circular bioeconomy some of the path we have already discussed. We are just taking some of the path which is not taken. So we are taking for replacing the some kind of the petrochemical input with the bio-based carbon neutral alternative. So now you can see that even government is also promoting the some kind of the biofuel, and this biofuel is generated from the agriculture waste, and from the agriculture waste you can generate the ethanol, and that ethanol can be blended with your normal petrol, and with it can be utilized. Similar to that, people are now also talking about the carbon capturing, because from each and every different kind of the process we are generating the lot of CO2, and that is basically responsible for the entire climate change. So carbon capturing systems are there. So now people are focusing on the microalgae and the cyanobacteria which can capture the CO2, and that can convert into some kind of the biomass which can be utilized by the different processes. So that will solve our two purpose. That will reduce the CO2, and at the same time only that will generate the different kind of the biomass that can be utilized for some construction, some food additives, or it can be also utilized for the biofuel and other. At the same time, our earth is getting contaminated with the lot of xenobiotic compounds and other. So the challenge is that we have to develop the different kind of the bioremediation and biodegradation technology by which you can just go for the entire part of that. And even similar to that, nitrogen fixation and other biofertilizer can be developed in this era only, right? So bio-based chemicals and materials. So whatever you talked, that was about the different kind of the agriculture, health and other, but now people are also focusing on the material which we are using, and people are asking for creating the material from the bioplastics. So you can have the bioplastic and biopolymer, and from that suppose something is created, then that can be reused, that can be degraded, and that will not create the problem like the xenobiotic which is created by the other. So you can have the polylactic acid by which the 3D bioprinting and other things are done. We are also using the lot of PPCP, pharmaceutical and personal care compound, which is taking the lot of surfactant and other, but now people are focusing on the biosurfactant, where instead of using this you can utilize the natural biosurfactant to that. Similar to that, you can have the lot of bio-based adhesive, you can have the bio-based solvent, pigments and other. You can see that in the Surat we are having the textile industry that is utilizing the lot of chemical different kind of the dyes and other. At the same time, in some part of the India like the Patan, Jaipur and other, some kind of the natural pigments are utilized. So now government is also looking for improving such kind of the pigments in the textile industries and other. And you can also have the biomaterials, so you can just go for the biogas, nanomaterial and lot of other kind of the material which can be utilized in the commercial purpose. So that is developed the entire new phase in which you can just have the bio-based chemicals and the bio-based material. So biomanufacturing, the bio-based economy should be developed. So already we are knowing that fermentation scale-up is done, and then you can just have the cell-cultivated foods, and even you can have the biosynthetic material out of that. So these are the few examples of the climate-resilient crop. Now there are a biggest challenge that whatever the climate is there, that is changing the entire pattern. So the monsoon pattern, temperature, everything is getting changed. So now we have to find out the climate-smart agriculture crop which can sustain in that environment, and in that environment only it can grow. Similar to that, you can also utilize the biopesticide and biofertilizer. At the same time, throughout the world, we are having the problem of the nutrition. So now we are just looking for some kind of the solutions like the golden rice, where the vitamin B12 is also, carotenoids is also produced by the rice and other. So you can have the biofortification of the some products. So that will solve your two purpose. Instead of producing the drugs in the different kind of the industries, you can take the food as a drug. So that is a way you can have the development in the agriculture, innovation and other parts. So some of the things already we have done. We can just go for the precision fermentation also, and that will transform the ingredient part. Similar to that, we are also having the bioenergy solutions. So currently we are in a 2G era only. So 2G, that is known as the second generation bioethanol production. Similar to that, suppose you are using the algae biofilm, that is coming in the 5th generation. 5th generation is the generation where we are utilizing the waste material. We are taking the CO2 directly from the nature. We are not disturbing anything, and from that only we are generating either hydrogen or some other kind of the energy source and other. So in that only the algal biofuel is coming, and even you can have the compressed biogas that is also developing right now. So the bioenergy solution, it should be also coming from the biology part. Genomics and personalized medicine, now slowly people are going for the biobanking. So there are so many things which cannot be edited by the human being, but human itself can become a source for the editing of that. So in the biobank, you can just store some kind of the genomes, you can store the stem cells, and from that only the different therapeutics can be converted. So that is also the era in which now people are working. Reliance has set up the stem cell entire storage center in the Ahmedabad and other. Now we are also talking about the CRISPR rewriting the code of the life. So now we are having the editing facility by which you can just go for the genome editing, and by which in future, currently also that is developed but not in a full scale, but you can have the some kind of the solution for the sickle cell disease, oncology, and even it can be utilized for the crop improvement and lot of other kind of the area. But of course that require the proper implementation of the CRISPR. Along with that, as I told earlier, that a lot of things are happening in the synthetic biology, but for the synthetic biology we should go for the design, build, test, and you have to learn that how it can be utilized for the other part. The AI and bioinformatics also is working. So along with that also the human microbiome, that is also a growing area. So a lot of things are normally managed by the microbiome. So if you are able to regenerate the microbiome, or if you are engineering the microbiome, some of the solution can be coming up. So some, even the startup is also coming in this era also, right? So these are the few area in which it is working. So now rapidly we'll be going through the different kind of the funding ecosystem in the India. So from state, suppose we are starting, then of course we are having the Gujarat state funding agency. So we are having the Guj Coast, we are having the GSBTM, we are having the government research aid for the faculty, and we are also having the great policy research. I'm not going in detail of all of one. You can get only the name and other things you can get from that only, right? So these are the few state level agency. The government is giving the Shodh fellowship. The GSB team is also giving the student support, and in Gujarat we are having the SSIP center at each university, and at the Gandhinagar we are having the iHub, where you can go with your prototype and you can have the huge amount of the funding and you can get the lot of support. So that support will be starting from the 2 to 10 lakh. So each university can support you up to the 2 lakh, but suppose you want to go above the 2 lakh, then you can go up to the 10 lakh at the iHub. Similar to that, you can have the iHub, and even the GSBTM has also developed the Biotech Park, so that is in the Savli Biotech, right? Besides that, we are also having the discipline-specific part, so we are having the GIRI, GCRI, and we are also having the so many other kind of the things where you can just go for that, right? So these are the different kind of the systems which are available in the Gujarat. Suppose at a national level we are going, then of course very common is now ANRF, so that is already available for us. Even the CSIR is also there, where also you can have the industrial research along with the CSIR lab and other. We are having the DRDO, that is doing the research in the different research and the development of the organisms in that. So there also you can work. Now the Ministry of Electronics and IT is also becoming very popular because they are giving the fund in the AI and other. So you can have the lot of digital or the AI project where you can work with the Ministry of the Electronics and IT, right? You can also have the DST, which is giving the different kind of the grants, starting from the sub up to the FIST and other, but now everything is covered under the ANRF form. Similar to that, we are also having the DBT, which is also giving a different kind of the grant, and some of the grant, like the BIREC and other, that is basically for the biotech startup. You can also have the BIPEP, you can just have the Biotechnology Industry Partnership Program, and same way you can also have the other kind of the funding from the DBT. We are also having the ICSSR, which is giving the funding for the social problems. So even a lot of problems are also society problem. So you can have the joint project with others from that. The IKS is also promoted in the NEP 2020, and you are also getting the grant of around 20 lakhs rupees in the IKS. And even IKS projects are also given. BIREC is a very popular scheme in this. So these are the few of the projects which are available at a national level. Now, similar to that, suppose you are going at a global level, then you can find out the lot of global opportunity. I have just sorted out few of 20 out of them. So I'll be just rapidly going through that only. So the biggest funding agency you can have, that is the NIH and NSF. So that is the American powerhouse. So a lot of people from India also, they can grant of the NIH. Even in Saurashtra University, people were sending their novel chemicals for searching for the anti-cancer activity into the NIH. So that way, you can also have the collaboration with them. Similar to that, you can have the Horizon Europe. So that is the world's largest multinational research program. So that is the European entire scheme in which you can just have the non-European country also as a partner. So you can work on that. So that also giving the different kind of the grant. We are having the Wellcome Trust and UKRI. So that is giving the UK Global Research Champion. Even in the GSBTM and the GCRI, they are getting the grant from the UKRI. So that is also giving the huge grant. Antimicrobial resistance project in the Gujarat was also funded by this. Similar to that, we are also having the Bill GITS Foundation. So that is also working on the vaccine, agriculture and global health equity. So that is mainly the area in which the biotech people can work. So that is also available. Similar to that, you can also get the other kind of the fellowship in the Europe and German, especially in the German. Suppose you are going, then you can find out that the higher education and research is vastly promoted. The only basic limitation is that you should learn the German language, right? Besides that, you can also have the fellowship in the Europe and other. So these are the USA Commonwealth Program, Fulbright Fellowship. So many such kind of the international collaborative research is also going on. So you can have the Indo-German or the Indo-EU kind of the Horizon program. So normally that is launched with the DBT and DST. So suppose you're having the some host in the foreign countries, then if such kind of the fellowships are coming, then you can go for that, right? So these are the few part of that in which the people are working, right? So this is a part which is normally required. Now suppose we are talking for the new generation, that is the biotechnologies, when you are just moving from your career into the era of the Bio E3 and Industry 5.0. Whatever you learn over here, that will not be sufficient. Of course, that will create a strong foundation. But besides that, you have to also learn so many new skills. And that skill will starting with the different part. So currently in the era of this, everything start from the thinking. And now the thinking also getting the design. So now people are going for the design thinking. So design thinking converts the needs into the demand. So you have to convert the entire scenario of the thinking, so you have to change the pattern of the thinking. So I'll take the very simple example of this. What you can observe in this is, there is a one simple story that one engineer was there, he has gone with the grandson in small village. His grandson, he has shown something like the swing, and after that when he came to the home, he tried to explain his grandfather that I want to have swing something like that only. He was not able to understand properly, but what engineer has done is, he has developed a different kind of the solution for making the swing. But ultimately you can see that at the last only, the children or the grandson was looking for a simple tire which is hanged with the one drop. So that was his need. But to reach that need only, the engineer has done so many kind of the things. So many a times solutions are quite simple, but we are not able to trap the real demand, and that's where the design thinking is coming. So now even for your PhD project or any kind of the things, you have to work on the design thinking. Now how the design thinking is working? So basically we are having the brain in a two part. We are having the left brain and we are having the right brain. The left brain, as we all know, that is analytical, rational, objective. It is like a scientist only. But suppose you are taking the right brain, that is a creative, holistic, so that is like an artist. But whenever you are going for creation of the product will always require both things together. So that will not require only the analytics, that will not require only the data, but that will also require the creativity. So when you are going further in the market, then you can see that always the person would be selecting the things by using the right brain. They're not using the things by the left brain. Suppose I'm asking you that which mobile is good for you, then what you will choose? You will choose the Apple. The Apple is good due to its design only. So that is the power of the design thinking, right? So the design thinking is normally changing the entire part. So you can have a traditional challenge where the scientific research often began with the materials methods, result discussion, and like that only you are starting with your PhD and you are also creating the project from that. But when you are coming to the design thinking problem, it will not start with the method. It will start first of all with the problems. So that is a basic part of that. So that is a design driven model. So that will start with the first part. So you can see that the first part always is the empathize. So there are two words, one is known as the sympathy and one is known as the empathy. Like that, for example, somebody is having accident in this room and they are getting the fracture in this area. Suppose somebody else is coming, then what they will do? That okay, you get the accident and that will be a normal sympathy. But suppose I'm asking that person, then what will happen? It would be empathy. Why? Because I also had that accident. I had the plate, I've removed the plate, I've gone through the entire experience. So when I'll be talking, I'll be taking my experience with him only. So that is basically known as the empathize. So always in the research, first step, what you have to do is, you have to empathize the problem, you have to take the user, and from that only you have to empathize that what is the actual need. From that you can go for the defining the problem, and after defining the problem only you have to go for the different kind of the part. So the empathize is normally using the behavior, environmental constraint, that is work on the real world constraint, and that will take the live experience. So depending upon all only the empathize would be done. The next part is that you can just go for the defining. So defining is a very important part. If you are able to define the problem is solved. Many a times it happens that even after completing the entire PhD, the students are not able to tell that what is the definition of the problem on which they were working. So you have to go for the risk of poor framing of that, and you should have the clarity in your defining part. And the problem framing, which is normally require the question like who suffers, where does the problem occurs, and why does it persist, and what challenge if it is not solved. Whenever the students are coming from the research project, they are telling that this we are doing because nobody has done. What is the reason that why nobody has done? There are two possibilities. One, that is worthless, or people have tried and people have kept as such only. So you have to find out that if that is the case, then why the people have failed and what actually is required. So that kind of the analysis you are supposed to do. After that, the next part is coming, that is known as the ideate. So this ideate now coming with a two phase. One phase is a researcher and another phase is coming from the empathy. So you've gone for the analysis of the user, you find out that what is required, what is failed, what is needed, and as a researcher you can tell that what you can create. So that will create a different kind of the idea, and you should create more than one idea for solving the problem. That is the ideate step, and after getting the idea you should go for the development of the small prototype. And again, instead of developing the one prototype, you have to develop multiple prototype, and always you have to go for the iteration on that. So you have to go for the testing, and after the testing you should go for the iteration in the research culture. So if you are following this culture, then you can find out that translation power of the design thinking is quite high, because that will solve the real problem, that will adapt the constraint, that will integrate the stakeholders, and that will accelerate the entire adaptation. And again, that will also attract the funding. So that is the reason that whenever you are writing the funding proposal, your funding proposal are failing due to the two things. One, it is not properly designed. Secondly, it is not having the technology readiness level which government is required. And even third, whatever the objectives are there, that is not aligned with the national or international priority. Right? So think like entrepreneurs. So whenever you are just working on the project, even for a simple thing, suppose you are starting working on a simple amylase, don't think that I'll be just completing the thesis and have the one paper. Think that what are the producers of the amylase in the world, and what are the biggest producer of the amylase, and how you can conquer that and you can become a producer of the amylase in the world. So that would be opportunity recognition. You can take the calculated risks and you can have the industry engagement and other. So these are the things I think you should go for the development. And even science is not a body of the knowledge, but it is the method of inquiry, a way of thinking, and above all, that is commitment to the better humanity. So I think that if you are changing the path, then future is the lab waiting to be built. So future of the India is also residing in your lab only. And if you are working in a better part, in future you can. Thank you very much for giving me the opportunity and listening my talk. Thank you very much.
And now I request Gaurav sir to please come forward, as we would like to felicitate our speaker, Rajesh Patel sir, Dr. Rajesh Patel sir. Let's hear a huge round of applause, please. And after this wonderful session, which was been taken by Dr. Rajesh Patel, hereby we come for a lunch break, but indeed making a very humble request to all of us that if we can complete it within 45-50 minutes, 50 minutes, okay. All right, so within 45-50 minutes, if we can complete our lunch break and once again gather for the next technical session, it would be indeed a great gratefulness towards all of us. So I am really hoping this from all of us, that quickly we can finish our lunch, take this break, use it, and come quickly back for the next session.